All right, I guess we should get started. Good morning, everyone. It's my great pleasure today to welcome attendees around the world to join BioVerse episode number 10. Sorry, not number 10, number 13. I already made a mistake to begin with because I'm too excited. And for those who are new to this, who don't know me, my name is Leon Tang. I'm the founder and host of BioVerse, which is a monthly webinar that discusses the most recent relevant topic in the global biotech industry, with a little bit of spin from the lens of Asian biotech companies and professionals. Let me share with you a little bit slides to tell you who we are. Just let me know whether you can see it. Thumb up, you can see it? Okay, so let me correct myself. This is episode number 13, the current R&D and deal-making trends in immunology and inflammation. Before that, like I said, I will introduce who is BioVerse, which is the co-production of 3 Asian life sciences professional-focused nonprofit, which are in Science We Trust community. We call ourselves I-Suite community. Second one is the Sino America Pharmaceutical Professionals Association with Philadelphia chapter, SAPA-GP. And the last one is the BioSpark Group, which is a Boston-based Chinese American majority life science nonprofit. Here, for those who are here the first time, please find us on LinkedIn. Just use a hashtag BioVerse. You'll find us. With that being said, it's my great honor today to have such an impressive panel of speakers to share their insights. Let me give you a little bit of introduction, each one of them. First, David Shen. Dr. Sam is a CEO and a founder of Proteologics, which is a Johnson & Johnson company. So David Shen is a well-recognized expert in the biologic drug discovery and development, and his leadership and Proteologics, a company he founded 3 years ago. Keep that in mind. Under his guidance, Proteologics made significant study in developing innovative drugs targeting I&I diseases. The company is focused on creating highly differentiated, multi-specific, and multifunctional antibody drugs aimed to enhance efficacy and extend molecule half-lives, thereby reducing the frequency of dosing for patients. His biochem goes very long, so I will make it a little bit short. So previously, David Shen held senior executive positions at NGM Biopharma, Teva, Amgen, and received his PhD from University of Toronto and postdoc training at MIT. Welcome, Dr. David Shen. Second person is Catherine Paulman. She has a PhD and MBA. She currently is a senior biotech analyst. So Catherine served as a senior analyst recently at the BTIG, one of the leading investment bank in the US, where she covers small to large cap biotech companies. Prior to BTIG, Dr. Paulman was a postdoctoral research associate at Weill Cornell Medical College, where she research focused on understanding the pathogenesis of different subtypes of prostate cancer, defining the molecular biology underlying the disease progression, and identifying new therapeutic targets. She earned her PhD in molecular and cellular biology from SUNY Downstate Medical Center, and a master's degree in biotechnology from New York University Tandon School of Engineering, and MBA from Berla Institute of Management Technology. Welcome, Catherine. Last but definitely not least, Dr. David Gu. Dr. David Gu is responsible for financial operation and financial strategy at Epimed. Previously, he was a senior analyst at Millennium Management, covering public investment in global pharmaceutical and biotechnology companies. Before that, Dr. Gu was a vice president of global pharmaceutical equity research analyst at Jefferies, where he was responsible for covering global large cap pharma companies across different disease areas. Before that, he was a consultant at McKinsey and also held equity research position at Wells Fargo. Dr. Gu got his PhD from University of Michigan and a bachelor degree in science from University of Illinois and Urbana-Champaign, UIUC. I never pronounced the name correctly. Sorry about that. With that being said, let's get into today's discussion. And obviously, a lot of people already heard a little bit of rumor. Well, a lot of rumors of what's going on this week. Pretty exciting. We heard some deals in the past, I would say 1 year, and a lot of breakthrough data release in immunology and inflammation. We call I&I space. Before we dive into a specific question, I want to give a little bit like overview of what's really going on. So I pull out a really quick table last night, and hopefully I get most of them right. I just look at what happened in the M&A space in the past 12 months. So exactly from August 9th, 2023 until 2 days ago. And what I found, first of all, a lot of M&A, which is good news, 50 M&A. And when I rank them, not surprising, we already have the sense, now there's a number. 20 out of 50 M&A in the past 12 months are in I&I. And not only the number is high, the number of M&A is high, the total amount of upfront payment is also pretty impressive. It's $30 billion US dollars. On average, each one of them is 1.5 billion. Now, if you break down into the 20 M&A in I&I space, you can say they're really very diverse, number one. Number two, most of major pharma already execute at least one M&A. Some of them did multiple of them. And today, I highlighted the two M&A, which, the insider, well, first one is a Proteologics, obviously, from David Shen. Another one, probably the freshest one, is from David Gu, the Vignetti Bio, who's like a lead asset actually come from David. So now people should understand why we invited the three speakers to come over today to share with their insights from R&D and deal making and investment perspective. Why this feels so hot? What's the nuances beneath those top line excitement, and what's the future going to be? With that being said, let's dive into the question we discussed. Oh, by the way, I want to emphasize one thing before we get into here. All four of us, the three speakers and myself, are iSuite community members. So I feel like this is kind of our daily conversation within our community, but this time we're going to talk this in public. Hopefully not as much restraint as we were in our community. With that being said, let's dive into the question. So the first question, I want to set the stage. I will stop sharing this as well. The first question I want to ask the whole panel is obviously, I&I is hot based on the data. There's so much M&A over there. And, but they are also different, right? So what's a key feature that set I&I space apart from other diseases? So if anyone want to, start, please feel free.
Yeah, I can get started. So for I&I, mainly the immunologists, that's the most important systems in our body. It's pretty much play a lot of roles, like certainly defense is the most important ones. And also maintaining this homeostasis within our body. It helps our body to heal some damages, and also mobilize our immune cells for different kind of injuries, infections. So with that, it pretty much involving all kind of diseases, as you know. Immuno-oncology, again, is the immune system, right? Metabolic, neuroscience, any kind of diseases has some roles of immunology play a role. So, and the other part is really like it plays a dual natures. It on one hand it protect our body, but if the homeostasis gets disrupted, then it causes damages to our body. So that's a very intricate balance of the system. And aside from that, compared to oncology, immunology or inflammation mostly is a chronic disease. like in the rheumatoid arthritis, lupus, Crohn's diseases. Those are chronic disease. Patients rely on the medicines for their lives. So therefore, the drugs need to be extremely safe so that people will not have side effects. And there's a lot of innovation, certainly in the immune system, the I&I, as you know, there's many validated targets such as TNF-alpha, interleukin-4, etc. Those are multi-billion or some like a 20 billion for the Humira used to be, and then the Dupixent now is catching up on that. Plus, the most recent, there's a lot of new innovations like TSLP, TL1A, and then most recently was a CAR-T to demonstrate like eliminate B cells for the lupus, for a lot of autoimmune disease. And then like this week or last week, BCMA CD3 engagers again shows very exciting data. So with all these kind of things, it definitely makes I&I pretty unique in this in our body system and also in the drug discovery process.
Wait, maybe I can kind of, I agree with David, whatever he said. And before that, good morning, everyone. Leon, thank you for inviting me. Glad to be here with David Shen and David Gu to discuss one of the most exciting areas of biotech. Like David said, I&I basically refers to immune-mediated inflammatory diseases that are caused by overactive immune system. It's heterogeneous like cancer. It can be limited to a specific organ or it can be a systemic disease. Chronic autoimmune diseases are an important part of I&I where immune cells become dysregulated, like David mentioned, disrupting homeostasis. And the immune cells, they attack self-antigens expressed on the surface of normal cells. And when we talk about the biology, there are a lot of factors that get involved, but especially in a lot of different types of immune diseases, autoantibody secreting B cells are a common factor and play a big role. They further interact with CD4 T cells through self-antigens, checkpoint receptors, cytokines for activation. So all these signaling proteins can be used as targets for new drugs. And we are going to discuss that more in detail later. But, just to give an example here, lupus or SLE, which is one of the key autoimmune diseases that is being targeted by this next wave of drug development. It's a systemic autoimmune disease. SLE patients, first thing, they produce autoantibodies against DNA, histones, nuclear protein, nucleolar RNA, and even HLA-E. And these autoantibodies lead to type 3 hypersensitive reaction in which these antigen antibody complexes, they activate complement system to a point that it can lead to organ damage. So clearly, complement system antibody producing B cells play an important role here, and these B cells need help of T helper cells for their activation, which involves cytokines like IL-10, IL-21, as well as CD40 and its ligands interaction. And then SLE patients also have depletion of NK cells from when we talk about the innate side of immune system. And again, they also have less Tregs, which cannot tune down the activity of T helper cells. So basically, with all this biology, there's so much to target to fight the disease. You can target B cells, you can target T helper cells, Tregs, IL-10, 21, CD40, and all these efforts are in the clinic right now. And the interesting part here is that when you compare the biology for drug development efforts, it's like a flip side of cancer. For example, in cancer, you focus on activating the immune system to fight cancer cells. In autoimmune disease, you're really suppressing the same immune system so it doesn't attack your own cells. And I guess that's why all the drug development efforts and technology advancements that have happened in oncology, it can be used for I&I, like, checkpoint inhibitors for cancer. They are used to block activity of checkpoints like PD-1 and CTLA-4, whereas BMS, abatacept, which is approved drug for rheumatoid arthritis, is designed to enhance the activity of CTLA-4. And then everybody knows about CAR-Ts and their success in cancer. All those efforts in the cancer space have led to development of better technology to design cell therapies that are now focusing on autoimmune diseases. And I guess we'll talk more about that later in this session. And I guess the last point I want to mention is that, the trend of moving to precision medicine, which started a while ago in cancer and is now being followed in autoimmune disease space, like KRAS inhibitors. For KRAS mutant cancer in autoimmune disease, we just saw like $11 billion acquisition of Prometheus Therapeutics by Merck that was developing an antibody targeting TNF like ligand 1A, which is involved in pathophysiology of inflammatory bowel disease. So a lot of these technological advancements can really help this next wave of therapeutics in I&I.
And I think I'll just chime in very quickly as well. So thank you very much, Leon, for the invitation. And I'm very happy to be representing IPMAC here on this panel. So, I think I've been very fortunate to be covering the large cap pharma space for like over a decade and just watching the evolution. So I think the question you're asking about with like I think is extremely interesting. But more broadly, if you look at all the key therapeutic areas, like oncology, like I, like antivirals, like the CV metabolics, all of these spaces have really seen, a lot of new biology and a lot of new mechanisms that have come out, and also the pharma companies that are willing to invest more to explore these mechanisms. So this is really we're seeing that explosion, in all these new treatments and new modalities. But I think more specifically also, I think for like I, one of the interesting things is that for a lot of the targets, I think it also has to do with biology as well. Because I think and I'm sure that Dr. Shin, he's been doing this for a very long time, but if you look at since it hasn't been that long really since the first monoclonal antibodies were actually approved from Genentech, right? It really hasn't been that long. But if you look at the entire like I&I space, for the majority of these targets, it's really also been like driven by this wave of antibodies, which I think is also very interesting. So, I'll just leave it there. I'm sure that, I think we still have a lot of other questions that we also need to cover.
Fantastic. That sounds like a multi-facet, like a advancement, right? Finally, they converge, and this kind of perfect time. Antibody, immunology, and probably also the willingness of the pharma company. Also, they figure out how to do this clinical, disease clinical trial. Great. So the second one probably get a little bit nuances like granularity. You mentioned that was a factor. Can you highlight a few, let's say, factors that really propel the current boom in the R&D and deal making activity in this space? Because we give a broad stroke, right? But now, what's a key factor if you can highlight a few of them? Do you think that lead to the R&D and the deal making? Catherine, you want to get started? I see you are nodding.
Sure. So, clearly I&I has performed well in the last couple of years. And before that also, it's a growing area. And we initially, started seeing a lot of venture capital interest in this space. And we've also seen now, a lot of equity offering collaborations and the M&A activity that you just showed, Leon. Besides that, Merck's $11 billion acquisition of Prometheus last year that I mentioned earlier. Takeda acquired Nimbus for about $6 billion. Iveric was also acquired by, I believe, Astellas last year for about $6 billion. For IPO, I&I accounted for 40% of the IPO money invested last year in biotech. This year we expect Zenas Bio IPO, which is around $650 million market cap. AstraZeneca's acquisition of Gracell that you highlighted in your present slide, for about $1 billion. And, these are just examples, some examples. So clearly a lot of interest, and there are a lot of multiple factors, I would say, that make the space interesting. And the first one is the market opportunity. For example, even autoimmune disease alone, there are more than 80 types, and about 5 to 10% of patients get affected by these diseases globally. So autoimmunity is about like $150 billion market. Now, there are some drugs already approved and used, of course, and they aren't really going anywhere. So the focus of these new drugs in pipeline is currently for more aggressive clinical manifestations or moderate to severe disease states. And that's also significant market because, like Dr. Shen mentioned, it's a chronic disease. So these patients take drugs for life, and the death rates are lower compared to cancer. So, that could provide a very attractive long-term revenue stream. And that combined with the fact that these drugs could be used for multiple diseases provides a multi-billion-dollar opportunity. AbbVie's Humira is a great example here, with a peak sales of about $20 billion. Another factor, I already touched on is like the technology advancements with all the efforts that we have seen in oncology can be used here. There's a relatively less experimentation required for something that's already approved. It makes it less risky to test it again. For example, you don't have to do R&D all over again to know CAR-Ts work well with 4-1BB and CD3s, or CARs work well with CD3s and 4-1BB. And another factor, in comparison with oncology is more favorable is the trial designs. Oncology, the standard of care is often, and so does the control arm benchmarks. For example, in non-small cell lung cancer, say, your control arm for second-line metastatic disease is going to be very different than first line. And even in first line, your standard of care options change depending on the PD-L1 status. But in I&I, like I said, majority of the focus is on moderate to severe diseases, in patients who have already gone through available treatment options. So the control arm here is placebo. And that makes a huge difference because it just simplifies the study design. It makes the data easy to understand and more reliable, including for investors. In oncology, nobody goes back to their animal models to see how their drug compares to different standard of care options that keep changing quickly, and that certainly is risky. And then the last thing I would say is like the commercial strategy. It's like oncology, even if the drug is not as potent as competitors, there's still room to develop it with differentiated commercial strategy. For example, the recent BTK inhibitor news from Sanofi, they really went after multiple sclerosis patients with secondary progressive disease and showed positive results, even when the drug didn't work well in relapsing disease. And some of the other companies like Biogen and Merck, they gave up on their BTK inhibitors. So, these are the factors that certainly make I&I a hot area for investment and, can drive significant value for shareholders.
Yeah, I would totally agree. It's kind of funny because you watch the AbbVie spinoff from Abbott back in 2012, and this whole space just completely taking off. One factor, as was mentioned, are all these new targets that are just out there, which is great for patients. You've got the JAKs, you've got your TYK2s, you've got the IL-23s, IL-17s, the TSLPs, you've got Dupi, IL-4, IL-13, you've got the IL-5s, OX40s. This has been a very, very incredible time in the I&I space in terms of the explorations of all these new targets. And then, with respect to the market opportunity, I couldn't agree more. AbbVie, they're so overweighted in this one therapeutic area because of Humira. And now they have to go to the JAKs. They have to go to the IL-17s. They have to go to all these new areas. And J&J, Amgen, Novartis, everybody, they're all going there. They have a lot of dollars there right now that's coming in, and that dollar has to be reinvested, on the pharma side. And this is a very high reward, but also lower risk, strategic area for them. And then also the other thing that you're seeing right now is that with these acquisitions, with these takeouts, for example, like Dr. Shin's, company, what you're now seeing is that all that dollar has to be recycled now. So you're also seeing all these VCs just raising, hundreds of millions or like billions of dollars, like, Foresight, JP Morgan Capital Life Sciences, Bain was just announced yesterday, 3 billion. There's so much money that is now flowing around, and so that's being recycled, but now they have to put that back in. So, I mean, where do you go? You go to the high risk and the high reward and the like low risk areas. And this is one of the ones that's the easiest to pick off.
Yeah, no, totally agree. Just to add a little bit more detail on that, you will see like scientific advancement. I think that's the key part of the I&I recently. This is because you will see like the successful TSLP, and then now you also see the FcRn and TL1A. There's a lot of excitement, including OX40 ligands, c-KIT, a lot of novel pathways being demonstrated in the clinical space. And also in my first part, I mentioned the key difference of this type of disease is the chronicity, and patients rely on those medicines for their lives. So with that, first of all, efficacy is very critical. You need to have a better efficacy because the old drugs like steroids, there's a lot of safety issues, and also efficacy not that great. So you will see that my company will use bispecific approach, try to bring two pathways to generate synergies. So that's really try to push the efficacy ceiling. So now you already see some data for that. And then the other very important aspect is really safety. These kind of drugs need to be so safe, not like in oncology, you can tolerate some safety side effects, but this type of disease, you cannot. So this is why like using antibody biologics actually play a very, very majority part of the drugs in this space, because biologics is a lot safer than small molecule. And now with convenience, right? Again, because it's a patient rely on those drugs for a long time. So you really need to have a convenience. The early part of antibody, you can do every 2 weeks or maybe monthly. Now we are pushing to every quarter or even every 6 months with the half-life extension. Really makes it very, very convenient for patients. So all these kind of factors really drive the I&I excitement recently.
Fantastic. Probably we can now, is a perfect segue to the third question. We dive a little bit into the scientific and the technology front. So, like Dr. Shen, Dr. Gu already mentioned a little bit, there's a lot of technological and also scientific, advancement. So just like narrowing on here. So probably we can start with the, we can focus on, which modality and which targets and which procedure you think right now is really gaining a lot of attention? Because I mean, the whole field is hot, but it's not like everyone feel the same like a love, right? There must be some kind of, there's always a favorable child or versus not so favorable child. So in terms of on the 3 dimension, right? The modality, target, and procedures, which one, in your opinion, really kind of standing out compared to others. Cameron, you want to take a try?
Sure. So, it's kind of obviously there, when you talk about modalities and targets, it kind of the combination of them is really important. And apparently CAR-Ts and bispecifics are getting a lot of attention. But, when you're talking about the first thing is like the disease areas also make a lot of difference to kind of decide where you want to go. There are certain diseases, those are less rare than others, like IBD, arthritis, lupus, and type 1 diabetes. So you see a lot of R&D efforts there. But again, anything that works in one type can also work in another rare type of disease, like Humira is a great example I mentioned earlier. And then, modalities, I guess I kind of like take them as like the overall approaches, and, and the recent success of multiple drugs and some M&A activity have clearly shown that, both types of drugs, whether they are completely novel or, based on novel modality or novel target, or are simply better versions of, validated targets, which is basically having an advanced technology, both could become blockbuster drugs. David Gu provided a lot of great examples. So I'm just putting things in context here, probably. But AbbVie's Skyrizi, which AbbVie believes could help offset Humira revenue erosion, is simply a better version of Tremfya and is administered less frequently. Both are IL-2 inhibitors. They bind to the same P19 subunit. And similarly, Apogee Therapeutics is developing IL-13 inhibitor, which has the same mechanism of action as Adbry from Leo Pharma's and Lilly's drug. And, they have shown a better half-life, so it can also provide dosing advantages. Similarly, MoonLake is developing IL-17, where 4 other agents are already approved. And then multiple companies are into alpha-4 beta-7 integrin inhibitors. Protagonist has an oral version where, which makes sense after seeing success from Takeda's drug, Entyvio, which is an IV version. And another one, Viridian's drug for thyroid eye disease. The stock was up yesterday about 30% after they reported data that showed at least comparable efficacy and safety to Horizon's Tepezza, but with less frequent dosing. So there's clearly a lot of potential to improve upon the targets that have already been approved. And then, novel approaches. For example, Abivax is developing interesting small molecule or oral therapy that can reduce inflammation and downregulate activity of Th17 cells, which play an important role in IBD. And, some of the examples earlier I gave, which is Prometheus, TL1 targeting drug. And then there are approaches, those are well validated and approved in cancer. For example, Anaptys Bio is developing PD-1 agonist for arthritis and ulcerative colitis. And we know, we all know PD-1 antagonist Keytruda, which is approved for several indications. Similarly, after getting monotherapy approval, T-cell engagers are now moving in this space. Cullinan has a CD19, CD3 bispecific. Xencor recently announced multiple bispecifics they plan to evaluate, including that TL1A IL-23 bispecific, and they have 2 T-cell engagers, one targets CD19 and the other one is CD20. And finally, cell therapies, which itself is a huge space. I believe we'll be discussing CAR-Ts in detail shortly, but there are a lot of different flavors of cell therapies as well. Besides CAR-Ts, we have NK, mesenchymal stem cells, as well as Treg therapy. And, I want to highlight a few approaches here. Starting with Tregs, these cells have impaired function in multiple autoimmune diseases, and the mechanism behind this impairment also differs with each disease. Like I mentioned earlier, in case of lupus, we see autoantibodies against HLE, which Tregs use to suppress activity of T helper cells that are responsible for autoantibody producing B cell activation. And in multiple sclerosis and myasthenia gravis, Tregs just lose their ability to migrate to the disease side or to provide their ability to provide suppressive signals. So engineered Tregs are being developed to address this concern, and it does sound vague when you theoretically think about it, but there's a lot of deals in that space as well. BMS had a $2 billion deal with GentiBio, which is a private company, and Regeneron had a deal with Sonoma Biotherapeutics, and AstraZeneca also had a deal with a private company called Quell, all in this Treg space for different autoimmune diseases. And another one is like Allogene is using a dual CAR, for their cell therapy, which targets both CD19 and CD70. And the overall idea of using CD70 is that it could obviate lymphodepletion steps, which seems to be a key hurdle for CAR-Ts. And lastly, on emerging therapeutics, there are multiple cell therapy companies trying to move away from auto CAR-Ts because it has manufacturing implications, but they are also trying to address graft rejection issue allo CAR-Ts could face. So CAR-NK, mesenchymal stem cells, they all fall in this category, and some companies are working towards these approaches like Fate, Nkarta, Century, Mesoblast, Cartesian. So, it's not just, one thing can just conquer everything. It's like cancer, you will get a lot of opportunities because there's a lot of heterogeneity. There are a lot of opportunities.
Yeah, totally agree. Yeah. So just to add on top of that, like if you look at the bispecific or multi-specific area, there's two major camps, like the T-cell engagers, this type of technology, we call it depletion technology. So you use the CD3 to find, or you use like the one arm to find a cell type, like the B cell using CD19s or the BCMA, and then the CD3 to bring T cell to pretty much eliminate those cell types. So that's a depletion technology. Plus the other approach that proteologics we use is we look for two pathways. Once you put them together, there's a synergy. So we call the 1+1 more than 2 approach, really try to push the efficacy ceilings, try to be the best in class in this kind of functions. So these are the tools are doing extremely well now. There's a lot of excitement in this area. So certainly that will generate a lot of new molecules, innovations in this space.
So Dr. Sen, actually, I
Yeah.
want to follow up on what you said real quick. You mentioned the bispecific. Obviously, the two lead assay from your company, both are bispecific, and one of them actually have a target TSLP, which you already know have multiple deals before your deal, right? So I remember, there's another one out of China. Henry got a TSLP, right? There's some intermediary, and then went to GSK 1.4 billion in total. I mean, 1 billion upfront, 400 million total value. But after that, your company get acquired, and your asset actually is preclinical. That asset was a phase 2, if I'm not mistaken. So that's actually really tied back to what Kavern have been saying. You don't need necessarily go after the novel target, the old target, right? You establish a target, but somehow you address some medical need. You still have a tremendous value in this space. I found that fascinating. Can you spend 1-2 minutes, give us a little bit of education? What's going on with TSLP? Why people put in so much money? Why people, in oncology, we always said you have to be top 3. If you're number 4, you have no chance. But this system doesn't apply to I&I, right? Probably you can educate us a little bit what's already going on. Just use that as a case study, if you may, a couple of minutes.
Yeah, so certainly. TSLP, as you know, Amgen's molecules generate a lot of excitement. The major advancement in this part is really in the T2 asthma, right? Because as you know, there's a lot of very good drugs like interleukin-5 and interleukin-4 receptor antibodies, or even IgEs. All these are kind of T2 asthma drugs. So they are targeting the Th2 pathways. And TSLP is the first time they target both the Th2 and the non-Th2 pathways. So that drug can treat T2 asthma and also the non-T2. So, and non-T2 is about 40% of the asthma patients. So you really have a much broader treatment for those inflammatory asthmas. So that's the a lot of excitement. So this is why it generates so much deals in the space. And when we try to look at the TSLP, we really look at why like Amgen failed in the atopic dermatitis. They did the clinical trials in atopic dermatitis, but it didn't work. And also Amgen did the steroid tapering for asthma patient, also failed in that clinical trial. And we spent a lot of time digging into their clinical data. And actually we find out like one of the key inflammatory pathways, which is IL-13. So in the clinical trial with TSLP, you will see IL-13 is reduced, but not bring to the basal lines. So still you have an elevated IL-13 in those patients. So this probably explains why it failed in atopic dermatitis, because IL-13 is the key player in the atopic dermatitis, and also in the steroid tapering process is also involved the IL-13 pathway. So this is how we initially proposed TSLP plus the downstream IL-13. If you block both pathways, then you will have a much better efficacy for that. And that's our story. And later on, we see Sanofi publish their data on that molecule. But when we proposed that, we didn't know Sanofi is working on that molecule. Their molecule actually through the acquisition. So that's the key story, because the differentiation and then 1 + 1 is more than 2, that kind of story really resonate with investors, with big pharmas. So this is why there's a lot of excitement for our molecule.
Yeah, we're gonna come back, ask you share the deal making and the investment cadence perspective. Before that, let's just let Dr. Gu also share your thoughts from scientific, R&D perspective. What's your thoughts over there?
Yeah, I would definitely leave the science to the true expert in the house today, which is Dr. Shin. But I think, hearing about the thought process and how he designed this compound, how you create a differentiated compound, I think this is exactly right. And, this is, this is how, this is a way that, new drug development should be done. So, I think this was an incredible asset and an incredible deal. But I think, one point that he touched on is exactly right, which is really about the synergistic effect that you see. And this is exactly why, for our company, so we focus on multi-specific assets. And right now, we made this strategic pivot a few years ago to go to T-cell engagers. And, I think your question on, like, what are the emerging areas? I mean, I'm probably biased, but I would say that T-cell engagers is really going to be the next hot area here, because you're really seeing the the ability for this modality to be applied, like not only to oncology, but now to like autoimmune, which is going to be incredible. And the basis, of this, which you take a step back, is you drag two cells and you put them next to each other to create some sort of either a killing effect or whatever. I mean, this could be applied to a range of cells, which is why, I mean, not only T cells, but you do see people also, applying this to potentially Tregs. I think, Kavari, she mentioned this before, but also to like macrophages, right? So I think, using this bispecific approach, I mean, TCE, I think it has the most amount of data, especially now that you see the oncology data with like Amgen for their DLL3, for their STEAP1, you see like Janux with the PSMA. So this is going to be, this is going to be hot. Now you also have this opportunity within autoimmune that people can explore. So I really think this is, from a risk reward perspective, it's exceptionally high risk, but also it's exceptionally high reward, but low risk. But, I think this does, I think this, for the companies that have the experience in this area, that have accumulated the experience, I think that would be important. So here at IPMab, and I have to give a shout out, is we're fortunate that we've already pushed 2 TCs into the clinic. Once the BCMA, which we have licensed out, but also the ROR1, which is also in the clinic. I would say that this right now is probably a first in class globally. And then we also have a few other exciting programs preclinically. So, the accumulation of that experience is extremely important. But, I think outside of the TCs personally, I still find PROTACs to be extremely interesting, this like protein degradation mechanism. And then also the oral peptides. I mean, you see Protagonists, you see DICE, I think you see the PCSK9 data from Merck. I think this is actually a very, very interesting space as well, from my perspective. So, I don't think it's just I&I, but you really see a lot of these new modalities out there. And, CAR Ts, I think we certainly hope that they will, that they can be used more broadly for the sake of patients. But from a competitive standpoint, we obviously, get asked a lot, the TC versus CAR T debate, but we can get to that later. And I think, with respect to disease area, this is also a very important question. At the end of the day, you have to look at the unmet needs, but you also have to look at the size of the opportunities. So it's really a balance of everything. So I'll just stop there.
Wonderful. I think right now, hopefully, I have already learned quite a bit, broad stroke picture of what's really hot from scientific point of view and also, kind of development point of view. Now let's just use two kind of recent deals as a kind of a case study, because both of you are heavily involved in both of them. I hope you can provide the insights more from a deal making and kind of a corporate development perspective. I would like to start with the Dr. David Shen, right, Proteologics. I'll give a little bit high level audience who don't know this, which I think you should. The company was founded about 3 years ago, and it was acquired, I believe, in May by Johnson & Johnson with $850 million upfront payment and with a $100 million milestone down the road. And both assets bispecific in identity space, and both of them are preclinical stage. So that's probably give you enough information why this is such an impressive deal. With that being said, I would like, David Shen, if you can share, I know that the journey looks short, but it's not as straightforward, right? There's a little bit detour. If you can share with us what you should really expect as a startup, or kind of if you do best-in-class drug, I think that would be very helpful to our audience.
Yeah, no, certainly it's a long story, right? So we started a company in July 2021 when the biotech markets start to drop very, very quickly. So And then after like 6 months getting to 2022, our investors telling me, say, David, the market is really going to like in a long time going to be very difficult to raise money. Why don't you try to raise some money so that you can last a lot longer because you have a very good story to tell. So we did try in 2022 try to raise some money by telling our very interesting story. And we talked to a lot of US investors, and majority of investors happy to listen, but pretty much just sit on the sideline until some momentum happen. So pretty much saying, okay, if you have some clinical data, talk to us again. But your story sounds interesting, but it's too early. So it's a very, very common kind of reactions. And we did convince one very top US investors, their lead investor and then their IC committee. And we were told that just one IC member voted against it, so it didn't go through. So I think there's certainly a lot of pain during that time. It's extremely difficult to raise money. And then the major reservation just saying, okay, TSLP failed in atopic dermatitis. Why you think your bispecific will work in atopic dermatitis are better than the other drug? So that's because there's some negative clinical data, so that makes investment very difficult. But like for a company to be acquired now with so many experience, so we really keep on telling people you really need to have a lot of stars aligned to make the deal happen. It's just like the stars eventually form a galaxy, then this is happen because you need a very top tier leadership teams with track records, very good scientific stories. Differentiation is so critical, and you need to have a very strong data to demonstrate your differentiation. And then the disease in the unmet medical needs, very big market, molecules pretty much ready for the clinical trials. And certainly the right momentum is so important. In 2022, the momentum is not there, right? The market is really in a very, very difficult situation. Nobody wants to invest at the time. So the momentum is critical. And then you really need to be ahead of the game. If you're way too behind, then you have no chance. So NIP, so there's so many things need to be aligned in order to make the deal work. So this is why we said it takes a lot of things to make, especially the acquisition of the whole company. We gone through like a fundraising. Actually, before the acquisition, we already have more than 150 million committed investments. We already have a term sheet. We gone through all the legal rules. So the money is almost there. And then because of the acquisition, we stopped the fundraising part. So all these things with our experience is acquisition is the most challenging, most difficult one because not just science, they really need to make sure the company has no issues on legals, no any kind of even EHS, all these things, they have to dig everything up to make sure that you are in very good state.
Wow, that's a fascinating story. Just any question from Cavalry and Dr. Gu before we move to Dr. Gu's story? No? Okay, now let's second case study. Again, I give a little bit of a prelude. what happened. So if you are living Mars of biotech, you probably didn't realize. If you are on Earth, you probably heard one of the largest fundraising in INN space announced this Monday. Candy Therapeutics launched with the $370 million led by Ken Song. And he launched with the pipeline, not very long, 2 assays. One of them is a BCMA targeted T-cell engager. Guess where did that come from? That come from David Go's company, actually. So David Go has been working with the Ken Song, and I look at the PR. There's probably 30 investors, right? So many investors. So it looks like you probably get more than half of the tier 1 investor in the US participate in this kind of a gigantic round. You know, it looks like a mind-boggling. Now, several things I really like David Go to give us a little bit insights. So initially, you spun off a new call last week called Valgeneta, or did I butcher the name, or did okay?
It's a Vignette.
Vignette. So you spun off the new call, and less in 1 week, that new call was acquired by Ken Song's Candy Therapeutics, become the backbone of his, one of the largest, kind of I&I launch in T-cell engager space. And also last week, there was a landmark paper, pretty much similar to your assay, the Take Valley from J&J, which is a BCMA T-cell engager, was tested in 4 patients and published in New England Journal of Medicine. Pretty impressive data. So 3 major things together, your new call, New England Journal paper, and this Monday, $370 million fundraising on the back of your assay. So tell us what's going on, what we can learn from this.
Sure. Thanks for the question, Leon. I think, to start off, I would have to say that Candid is really about Ken, Arvind and his team. I think he has shared publicly that this was a multi-step transaction with multiple rounds of fundraising. So, I would have to say that, we're incredibly grateful and happy that this asset is in the hands of Ken and his team. And we're extremely happy that, our investors really supported us throughout this process. But, having worked with Ken and his team throughout this process really makes me understand why he is, one of the most incredible businessmen and also biotech leaders. The execution and the strategic vision that we've seen from them is just incredible. But I would also have to say that this deal itself, more specifically with respect to our BCMA CD3 on Vignette, I mean, this was only 1 step in this whole process, and this was really done with the help of the investor syndicate, led by Foresight. But also, I think this deal itself, it was incredibly difficult, the entire process. So I think operationally speaking and, deal-wise, it was truly a team effort on our side. So, our CEO, Dr. Chunbin Wu, he really oversaw this entire process and, guided us. Our CBO, he was very busy negotiating. Our CMO, I mean, it's all about the data. And me, with the fundraising and the investors, that's also another hurdle. But I think it really comes down to the data. That's the most important thing, because we presented our data at the as a late breaker in 2023 SITC conference in October. So I was there in San Diego. We actually had a lot of people coming up to our poster to see the phase 1 dose escalation data that we had in multiple myeloma. And the key about that data, which I was emphasizing, was really the optimal design of our compound, which gave an extremely high potency that was, at that time, it was 87%. But since then, since we've gotten more data, it's gotten even higher. And also the fact that at the doses where we saw the strong efficacy, at the lowest dose, we had no CRS whatsoever. So, this goes back to what Dr. Shen was saying earlier, is that for I&I, you need a drug that is extremely safe, that's well designed. So this is actually one of the key things about our T-cell engager platform that we've been able to achieve. So this is what really attracted a lot of people to us at the JP Morgan conference. So it just became a very competitive process after that. You know, we were very fortunate to get a lot of TSs there. So, you know, after that point, it was really about, it's really about the step-by-step negotiation and then trying to figure out what is the best terms that our investors, especially our board, can accept. And also, you know, just executing on this licensing deal, but also the spinoff and the subsequent, you know, financing for our round, which we were fortunate to have Foresight, Samsara, Qiming, and also Mirai. So, but then the step after that for Vignette and also Candid, it's probably not the best thing for me to comment on. I would say, you know, I think this is probably best for Ken and his team. But, you know, I think there's a lot of articles out there. But what I can say is that the level of execution I saw from his team is just mind-boggling. I mean, I could see why he's extremely successful. And this is what you need right now, because overall, you know, I think if you step back, you know, there's a lot of CD20 CD3s, there's a lot of BCMA CD3s, there's a lot of CD19 CD3s. So the question that you have to ask yourself is, you know, who's going to win, right? So I think one thing that we do see is, you know, you have to have the dry powder, you have to have a very strong management team, and you have to have the strong execution. So, you know, I think, you know, this is why we are incredibly grateful about, you know, about this partnership between us and Candid. So I'll just leave it there.
Yeah, wonderful. Any comments before I can share one quick comment? I want like Kevin and Dr. Shin. You have anything you want to add after? Now, no, largely the set of story from Dr. David Gu.
No, I just want to say quickly that, you know, I completely agree. Bispecifics have made a lot of progress. They have a lot of potential. And, you know, after seeing Schett cell therapy data, it kind of gives you a lot of confidence that bispecifics can really do better than some of the other approaches like BTK inhibitors, BAFF/APRIL, or, you know, the belimumab, which is already approved. And all these like, you know, approaches that can deplete B cells can clearly be more powerful and have potential to provide cure over something like, you know, even monoclonal antibodies like rituximab or Gazyva don't really have that kind of potential.
Yeah, Kevin, before I let you go, I know you can pronounce Dr. Skid's full name the right way. Can you please educate us? Because the last time we met, you told me how to pronounce his full name the right way.
Oh, that's like just my assumption. I thought it was Jorge Schett.
Jorge Schett, you see? I know you know someone speak German, so that's why I trust you. All right, so that's fantastic conversation. We run a little bit over time, but I still want to use a little bit time. We do see one question from the audience. It's a really quick question. The question is, what opportunity do you see for anti-47 monoclonal antibody to get into autoimmune diseases? Thank you. I guess that's like a target macrophage, right? Any idea?
Yeah, no comments from me.
No comments.
Yeah, I will leave it to Dr. Shin, the true scientist in the room.
Well, we haven't looked into that one. Certainly, it's a interesting one, right? If you somehow failed in the oncology space where they repurpose for the autoimmune, it's certainly too early to tell.
Yeah.
I agree. You know, it's too early. You can't make anything, especially when it didn't work out in oncology. But, you know, just kind of like speaking of the biology. So CD47, you're really trying to bind anti-CD47 antibody is really going to bind CD47 so macrophages can see the immune cells that they can destroy. You know, it's like, you know, NK cells being depleted in SLE, for example. If you somehow can activate NK cells, they can perform ADCC and kill auto-antibody generating B cells. Similarly, if you activate macrophages somehow, you can kill these auto-antibody producing B cells as well. They can be recognized. But the question again comes out from a therapeutic index. How do you identify B cells, specific to autoantibodies versus something that's specific or they're just like normal healthy B cells. And CD47 is not something that is only going to recognize B cells. Like Gilead showed, it can recognize every single thing, including red blood cells, and get a lot of anemia. So, and in oncology, there was certain ways you can just develop therapeutic index, which hasn't been tested in autoimmune diseases. So, going back to what Dr. Shan said, it's too early to kind of come to any conclusion whether they can work really well, but there certainly you will need a lot of work in that space to figure out how to make them work.
I have kind of one quick comment. So actually, I was trained in immunology, but I'm not a very good one. I still remember when I was taking my immunology class, one of my professors told me, use a metaphor, he's a macrophage is a garbage collector. So obviously, when we try to educate those garbage collector in immuno-oncology, didn't work that well. They said whether there's a garbage collector become smarter in autoimmune disease and do something. But keep in mind, they're garbage collector. That's how people look at them. They're not like kind of the how to say cell 6, those special, which is T cell, right? That's a pretty high power, the one versus 1,000, no problem. They're just a garbage collector. With that being said, I really enjoyed the conversation today, but we do have a few kind of a quick announcement before we let everyone go. And let me just do that. And let me just also share the screen and get everyone's, just stay with me for probably 2 minutes. I want to first give a shout out of we actually have another panel discussion. If you happen to be in Boston this Saturday, please stop by. They're going to be the first annual conference from our collaborator, also one of the 3 organizer of this webinar, BioSpark in Kendall Square, epicenter of biotech. Please feel free, scan the QR code and join us if you have time. Last but definitely not the least, now, I hope you already like what we are doing. If you really want to learn more about us, iSuite community, please scan the QR code. You can learn the 3 major things we do. You already saw the BioVerse webinar. We also have a newsletter. We also have a lot of WeChat group and a very nice LinkedIn page. We have a lot of discussion over there. So scan the QR code and learn more about iSuite community. Last but definitely not the least, if you are enjoying what you are doing, what you are listening today, and you want to advertise your company, bring visibility, either as a service provider, VC fund, company, whoever, as long as in life science industry, please consider sponsor us. We would highly appreciate that. And our email is in the bottom of the slides, and we still have 4 fantastic episodes on a specific date and a very relevant topic already decided. And we have a very nice speakers. If you contact me or other volunteers, we are very happy to disclose to you all those fantastic speakers and a very interesting agenda. With that being said, thank you, Catherine. Thank you, Dr. David Shen and Dr. Gu, and thank you everyone who dialed in today. I hope you enjoyed the conversation and discussion today. We'll see you next time, and enjoy the rest of your week. Take care. Bye for now.
Bye-bye.
Thank you very much. Bye-bye.
Bye.